Why don't we go to the next chapter here? It is subdivided into two parts, motivation coming up first and then emotion. And sometimes we might pause to think, why do we stick motivation and emotion together into the same chapter, which is common to do in even in introductory level courses. And that's because a lot of the same nervous system structures are involved in regulating both of them. In particular, at the level of the brain, we're going to be looking at the limbic system, which includes the hypothalamus, but of course, amygdala and other aspects of the limbic cortex are all involved in the control of emotion. And also the fact that the peripheral nervous system, in terms of the sympathetic and parasympathetic nervous system, are also relevant here as well, because certainly on the motivation side of things, we're going to be talking about some of the basic physiological motivating behaviors like hunger, for example, and that is a part of the parasympathetic activation. But on the emotion side of things, when we think about fear and aggression and other things that we might associate with the fight or flight response, that involves the sympathetic nervous system. So we have sort of a contrast here between the kinds of things associated with parasympathetic activation versus sympathetic activation. But let's first talk about motivation here. So notice that I have it focused on ingestive behavior. I'm going to talk about eating for the most part in the details of this. But before we do, let's just talk some broader terms about motivation theory. The idea of motivation theory focuses on the concept of need. The idea here, of course, is that a human or an animal has needs, and when those needs are not being met, this creates some sort of a state in the organism, a state that is designed to somehow create behaviors. The behaviors, of course, now are designed to satisfy those needs in some way, all right? In the case of hunger, it might be fairly straightforward, right? There are physiological mechanisms involved in the energy needs of the body. And when we see that disrupted, we begin to feel hungry. That feeling of hunger then motivates us to go in search of food. So again, the behavior at a higher level psychological level seems disconnected from those lower level physiological states that are involved with our body's energy regulation. But certainly there's a driving force there. That concept of a drive is an important part of the biological drive theory or motivation theory. Just like we use the analogy here, think of a person getting in a car, you're driving the car, and that means you're telling it what to do, right? You're telling it to go fast or go slow or turn left, turn right, based on your inputs to the car. And in this way, some of these lower level biological processes are driving us, right? They're telling us to turn left and turn right. That is to go out there and find food in the environment. So the concept of a biological drive is part of this larger motivation theory. But motivations can be, highly varied. For example, we could talk about the fact that our bodies apparently need sensory stimulation, and we are motivated to seek out stimulation in the environment. We see evidence of this because of back in the 1950s and even a little bit earlier than that in the 40s, it was a lot of research that looked at sensory deprivation research. And we saw a little bit about this previously. We've talked about development, and we've talked about animals raised in deprived environments show less brain development. And from that, we get Hebb's law. We have Donald Hebb again here because Hebb was amongst those researchers who were studying these sensory deprived environments. He and Butler and Harlow had done some of this kind of stuff as well. And Harlow had also done this with monkeys as well, and showed, of course, that certain deprivations in infancy and adolescence in monkeys are associated with some declines of brain development. And we can do this with humans. Now, when we do it with humans, obviously we're not keeping infants and children in deprived environments intentionally, though there are certainly, of course, a few case studies of children who have been raised in extreme deprived environments, such as the story of Jeannie, who was not exposed to language for the first 12 or 13 years of her life. But Hebb has done other things with sensory deprivation research in adults, where he basically just, this whole concept, if you've ever heard of a sensory deprivation tank, that was something developed by Hebb, just to see how do people respond in these kinds of environments where there's really no external stimulation whatsoever. And one of the things that Hebb found is that people really dislike it a lot. And it's a little bit similar. You may have heard of some of these rooms that are called the quietest room in the world. These are called anechoic chambers because the walls are lined with a very strong sound absorbing material so that there are no echoes, and the room is very, very quiet. And so if you go in there, you can't really hear any sound. There's just no ambient sound that you would normally hear in a regular room. And over time, it gets to the point where you can like hear your own blood pulsing through your veins and stuff like that. But the idea is that sometimes people report that these rooms drive them crazy, right? They just hate it. And this idea that kind of being in this sensationless environment is highly unpleasant seems to suggest that maybe this is a need we have. We just, having some lack of external sensory stimulation is very unpleasant. So we are just motivated to seek out sensory experiences. This is actually even studied sometimes as a personality trait. There's a personality scale known as the sensation seeking scale, right? And the idea here is that some people score high on this. They are high sensation seekers. So that means they have a high need for these sensory experiences. These are kinds of people who like to go skydiving and engage in all kinds of other really thrilling and exciting things because they want that really high stimulation. Others score low on this trait, suggesting maybe they're not quite so interested in it, but they still obviously experience some degree of sensory input just in regular everyday life. When we talk about needs, we can talk about the biological things we might have bring up here, but we could talk about other needs, psychological needs, for example. There's these ideas, for example, there's something called self-determination theory that tells us that all humans, at least in theory, have a need to feel autonomous, a need to feel competent, and a need to feel connected to others. And so in that case, we could have a psychological theory of motivation that explains that certain people make certain choices in order to satisfy those needs for autonomy, competence, and connectedness. We might also even have emotional needs, right? We want to feel loved by others and seek out the company of others. But in the context of this particular class, we're not really going to get much into some of those psychological needs, and we're instead going to focus more on the biological and physiological side of things, hence that we are the physiological side course here. So we're going to start by talking about biological drive theory. So the logic behind biological drive theory is that when a need is not being met, as I mentioned earlier, this creates a particular state in the body. And we're going to call that state some form of an arousal state. Arousal states are sometimes labeled in what's called a negative valence. That is, it doesn't feel good to experience this kind of arousal state. Hunger might be an arousal state, and it doesn't feel good to be hungry. And so now I am motivated, to make that arousal go away, to make that hunger go away. And that returns my body back to a calm, balanced or state, which we call homeostasis, right? So in that case, I could describe, for example, a cat hunting to satisfy its hunger needs, right? So we might suggest now that this idea that eating when you're hungry is somehow pleasurable, right? Why is it pleasurable? Well, of course, because it may be pleasurable to reduce the negative arousal of hunger. But of course, it's also the case that sometimes these kinds of need-based behaviors activate the dopamine pathways in the brain. Satisfying needs and experiencing rewards are pleasurable because they also activate dopamine. It might also then explain why, for example, that certain behaviors are considered species-specific behaviors that are hardwired into our brains, because maybe it's evolutionarily advantageous for us to do so, but why is it? But why do we are motivated to do it? Because it might also be pleasurable. This is one way we could talk perhaps even about like sexual behavior and reproduction. It's evolutionarily advantageous for animals to reproduce. And at least in humans, we know that reproduction and sexual activity can be pleasurable. And so we might seek out that behavior because we might be engaged in some pleasure seeking on the part of that. But we might, in a sense, somewhat be slaves to our biology here, that evolution has made sex pleasurable to encourage us to do more of it, because doing more of it means having more offspring, and that means our genes are passed on to future generations, ensuring the survival of our genetic lineage and ensuring, broadly speaking, the survival of the species, right? And that means then that once we start talking about instinctual level behavior, we can talk about a variety of different things that activate those behaviors. So the concept of a releaser stimulus is the environmental stimulus that triggers the behavior. This is at least very much the case in many animal species. When we talked about fish mating previously, the stickleback fish, right, that the appearance of the female with a swollen belly full of eggs is a releaser stimulus that triggers a mating dance from the male. All right, and so again, it just kind of triggers these kinds of behaviors. Now, is it really the case that humans are that instinctual when it comes to sexuality? And the answer to that is, yeah, maybe a little bit. There's a lot else going on too, because we certainly have a lot of psychological factors at work with regard to sexuality, not to mention a whole host of sociocultural practices as well. And so all of these things will factor into human behavior, not purely just biology, but certainly worth listing here. And so, of course, part of that could be when we talk about the releaser stimuli with regard to sexuality, maybe we can think about olfaction. We haven't talked about this sense very much, and we're not really going to get into the details of how the olfactory epithelium works. We know that there are receptors embedded within the sinus that are part of what's called the olfactory epithelium. These are neurons that, kind of like the taste buds for taste, are chemical receptors. And so just like there are receptors on dendrites of neurons that are unlocked by neurotransmitters that have a specific molecular shape, the taste and smell receptors are unlocked by specific molecular shapes. But in this case, it's not from neurotransmitters, it's from things that we inhale, right? And the molecules of that chemical that we inhale will activate certain classes of receptors in the nose that triggers signals from those nerves that then travel to various brain structures. There is an olfactory bulb in the cortex, but there's also connections to the limbic system, such as the amygdala. So, for example, there might be some smells that trigger an emotional response right away, right? That could activate fear and aggression. There may be some smells that particularly might be the case in some animals where there might be territorial behaviors between males, for example, and dogs may mark territory with urine, or other animals may mark territory with urine. And then that particular smell kind of may activate different reactions from other males of that species, right? But there's other connections that go to the thalamus and then to the frontal lobe of the brain that help process the conscious perception of smell. But there's also, as part of the connection to the limbic system, there can also be, and this includes the hypothalamus here, connections that trigger mating behavior as well, right? So this is referred to as pheromones, right? And many animal species use pheromones during mating, right? So non-human mammals have a reproductive cycle in females called estrus. And this occurs on a seasonal basis. And generally, the females of those animal species are only sexually receptive during their estrus cycle, and they release pheromones that then trigger mating behaviors in the males. So again, in this case, then that is an instinctual response, that the mating response is triggered by the smell of the pheromones. There's a whole lot less evidence that humans use pheromones during matings. We do produce hormones that can alter our body's smell, and that may influence sexual behavior mildly. As I said, there's a variety of psychological and other factors. Excuse me. A variety of psychological and cultural factors that also influence that. So it seems that really humans don't really produce pheromones that regulate sexual reactions in the same way that they occur in non-human animal species. Taste, something similar could happen here. So we could talk about eating behaviors being triggered by the activation of receptors in the taste buds. So here, this is gustation, right? So we know that, as I mentioned, the receptors for smell are chemoreceptors, chemical receptors. And in this case, the same is true for the taste buds on the tongue. It seems that we have 5 primary taste receptors that register different so-called primary taste sensations of sweet, sour, salty, bitter, and umami or savory. The claim here, when it comes to explaining flavor perception, is a bit of a challenge. We might try to make a claim that the taste of something unique, like the taste of chocolate versus the taste of an apple versus the taste of a steak, I guess, is some unique combination of these 5 primary taste sensations and is therefore encoded into the taste nerves as some sort of unique balance of the 5 primary tastes, in the same way that our perception of color, for example, is thought to be some balance of activation of the 3 types of cones, right? As a primary color system, this would be a primary taste system. But in the context of the behaviors, we might suggest that here, again, the nerves from the mouth travel through to the thalamus. Some of it goes to the somatosensory cortex and the parietal lobe, which is that where taste is registered? Well, not quite, a little bit, but what's also happening there is that we actually have a lot of sense of touch within our mouths as well. And when we're eating, we can detect the texture of the food and whether it's crunchy and soft and so forth. And so there's some of that kind of tactile feedback that goes to the somatosensory cortex. Other pathways, again, like with smell, go to the hypothalamus and go to the amygdala, and could be, again, primary drivers of certain kind of instinctual behaviors, in this case, eating behaviors. Now, how does that work? Well, there are foods that taste good, right? And so again, this idea that certain foods taste good, why do they taste good? Why does sugar taste good, for example? Perhaps it's because we need sugar, right? And so the idea is that having some degree of sugar and carbohydrate in your diet is important to maintain blood glucose levels where we want them to be. And we may have an increased need for sugar when blood glucose levels goes down, and that might increase our desire to eat sweet things. So this creates a motivation that's very targeted. It's not just I feel hungry, but I want specifically something sweet. It might also be the case that in other situations, you might crave something salty instead, because when your blood pressure drops, your blood volume is dropping and you need not just water, but you need electrolytes, right? So salt helps raise your blood pressure. And so the idea is that sometimes we might, in addition to being motivated to drink some liquid, right, we also might be motivated to consume something that contains some salt content or mineral content that will help raise blood pressure as well. This is, of course, exactly what sports drinks do, because they contain both sugar and water and salt together, and that helps sort of increase your energy levels as well as replenish blood pressure and fluids in the vascular system. So, but this idea, though, that eating is pleasurable is the same as what I was talking about with the concept of sex being pleasurable. There's a biological reason for us to need to engage in those behaviors, but nature has equipped us with the aspect of making it pleasurable to motivate us to do it more often. And so there's the general concept of that, as well as the targeted idea that sometimes we may crave different things based on something that our body might be missing. So if we're going to describe motivations in these kinds of biological evolutionary ways, we need an explanation for how that works. And so we're going to talk about this concept of regulatory behaviors. What we're also going to see a little bit later in these notes is that there are also examples of other biological behaviors that may not be exactly regulatory in nature. But regulatory behaviors are examples of where we're trying to maintain a certain state of affairs, and that requires regulation. So an example of this is this concept of homeostasis that I also have in the slide. Homeostasis could be defined in a way, I don't like the term balance because balance implies some sort of an equalization of two things. Equilibrium kind of, again, difficult to do, but really we're just talking about maintaining a state that is predefined in some way as being necessary. So, for example, I was talking about drinking, right? So when you become dehydrated, your blood pressure drops and your blood volume declines. And so we need a way of regulating your blood pressure so that your blood pressure doesn't drop to dangerous levels, because if your blood pressure does drop to a dangerously low level, you can experience cardiovascular collapse, and then you lose consciousness and you can die. And of course, if you have really, really high blood pressure, you can have all kinds of other damage to the cardiovascular system, and you can have other health problems that as a result, consequence of that can cause heart failure, things of that nature. So we need a medium value here, right? Not too high, not too low. But how do we regulate that? Well, of course, in order to regulate anything, you have to have a system that can detect deviations from the desired value. So what's our desired value? Well, there's a range of what doctors consider to be healthy blood pressures. We could go with something like 110/70 is a pretty decent healthy blood pressure, right? Well, in that case, if the blood pressure gets too low and you're becoming dehydrated, your blood volume is declining, you have to have a way of detecting that, right? So we would call this a deviation away from homeostasis because it's not in the desired range anymore. And if it's too high, again, we would have to have a way of detecting that. And then once we detect that deviation, we have to have a way of correcting it, right? So how do you detect low blood volume and low blood pressure? As it turns out, we just got done talking about the somatosensory stuff before. There is a process of interoception, right? Perceiving the internal body states of our organs, and that includes our blood vessels. So there are stretch receptors, just like there are stretch receptors in the skin, muscles, and tendons. There are stretch receptors in the blood vessels. And when the blood volume decreases, these stretch receptors detect the contraction of the blood vessels. And that then sends a signal to the brain, such as the hypothalamus, the pituitary gland, and that activate the feeling of thirsty, right? Now I feel thirsty. That also becomes something that travels through the thalamus into the frontal lobe so that I become consciously aware I feel thirsty. This now creates the motivation, the arousal state to say, that doesn't feel good. And now I am motivated to go in search of water to drink, right? So here we are trying to maintain our blood pressure and salt levels to maintain the appropriate blood levels, right? The same goes for body temperature even, right? Like we maintain 98.6 degrees in our body. When it drops, we feel cold. This causes some physiological response. We get goosebumps, we shiver. These will do something to help raise our body temperature a little bit. But beyond a certain point, that doesn't do enough. And we become then consciously motivated to put on a coat or go find a warm environment to be in, right? So again, that's where the motivation side comes in. So all of these things are involved with that, with eating as well. We're trying to maintain energy reserves in the body. Waste elimination, again, there's a negative arousal state associated with having a full bladder or full bowel. And again, so relieving that through bowel movements and urination, these are all part of this regulatory behavior, right? I have an example here of a regulatory system, and it's a thermostat. So just like we could talk about maintaining body temperature at 98.6 degrees, here a house maintains a temperature, whatever we want to set our thermostat at, 19 degrees Celsius perhaps, okay. And so you have to have, as I said before, you have to have a detector. You have to have a sensor that detects any deviation away from your set point. The set point is where you have things set. If the temperature gets too hot in the house, you've deviated from the set point. So what's our detector here? Well, it's simply a thermometer. Thermometer detects the difference now between the current temperature and the set point temperature. And once that goes positive, like +3 degrees, where it's now 22 degrees Celsius inside the house, it's time to kick on the air conditioner, which lowers the temperature. So this is called a negative feedback loop, because what we're doing is we're using the information to feed back into the system to regulate it, but it's called negative feedback because the behavior of the system always negates the error. So when the temperature gets too hot, that's a positive error. And then we're going to cool things down by lowering the temperature. So we're negating the error to take it back to zero, right? Likewise, in the winter, when the house gets cold, we warm the house. So when the temperature, now the error signal is negative, and then we raise it back up. So we're negating the error to try to return the error signal back to 0 to keep the house at the set point. So again, we might think that maintaining body temperature works in the same way, having some sort of heating and cooling systems. Of course, we do, as I said, shivering and goosebumps, and then motivations to put on a coat when we're cold, but also when we're hot, we sweat. And then beyond the physical side of that, the sweating, we become motivated to find a cooler environment, to turn on the AC or go inside or jump in a pool or do something to cool off, right? But there's a lot of these kinds of regulatory systems in the body besides temperature, right? So as I said, blood pressure regulation, eating behaviors, those are all regulatory things. As I said, we also may have some non-regulatory behaviors. So in some cases, some of these kinds of behaviors are perhaps physiologically influenced and biologically influenced, but they don't necessarily involve specific states that we're trying to maintain. So as I had mentioned before, with sexual behavior, we may see some degree of sexual responsiveness influenced by fluctuations in sex hormone levels, especially in humans, more so than other animals, as I said. But I also said there's clearly a whole lot more cognitive involvement, psychological and cultural factors at work here. So that means the frontal lobes are doing as much, if not more work when it comes to regulating sexual behavior than, say, the hypothalamus and the limbic system and all those other parts. But what's also important here is that even if we acknowledge that there is a strong hormonal component to sexual behavior, it's not as if we're trying to regulate sex hormones at some level, right? That when the sex hormones get too high, I'm motivated to lower them. And when they get too low, I'm motivated to increase them. We don't have any good direct evidence that we're trying to maintain a homeostatic level of testosterone or estrogen somewhere in the body, right? So that's not really a regulatory process. It may be strongly physiologically influenced, but it's not regulatory because we're not regulating the sex hormones at some specific point. The same goes for all these others in this list here, right? Parental behaviors, again, because I said before that evolution wants us to reproduce and have children, we are motivated to engage in reproductive behaviors, but we're also motivated to care for our children to ensure their survival. That again may have a strong physiological component, but it is not about regulating any state of affairs, right? Same goes for aggression. Food preference is iffy. So I'm kind of contradicting myself maybe a little bit with this because I was suggesting before that sometimes we might crave something sweet or salty because of some deficiency in our body. So that might be regulatory, but other things, just about liking certain styles of food, for example, if your favorite food is pasta with tomato sauce or something like that, that kind of level of preference, it cannot be explainable in those kinds of regulatory terms, right? It's just much more of a sensory preference there, right? It's just like preferring the color green over the color blue, right? It's just what you like. So in general, then we have these kinds of ideas that could have physiological elements to them, right? But are also much more strongly cognitively influenced. I have this final little distinction here of intentionality versus instinctuality. It's an interesting way to break this down a little bit, where the idea is instinctuality is where we might talk more about animal behaviors, where the behaviors are much more limited to pure instinct and physiological process, whereas for humans, that oftentimes is overridden by a variety of cognitive goals and social goals and cultural norms and other things that we also are expected to follow. Right. But let's talk specifically about the regulation of eating behaviors, right? What's the regulatory involvement here? So first, a few brain signals here. We have the brainstem, we have the medulla. This is part of the autonomic nervous system and parasympathetic nervous system. That's just going to keep all of those systems functioning all of the time, right? But one of the things that it does is it helps detect any deviation from our desired state. So if there's a hunger signal arising from the body, that signal is going to be detected by the medulla. If there is a satiety signal, so when you feel hungry versus when you feel satiated or feel full, again, the medulla is detecting that. There are also those interoceptive signals. So when your stomach is empty, right, that's a part of interoception. You can feel your stomach empty and grumbling. That's detected in the brainstem, or when it feels full. And actually, the same could be said also for your intestines. We're not necessarily always consciously aware of when the intestines are full of food, but certainly that is registered by the medulla anyway. And the gustatory input. Now, what's interesting here about the gustatory input to the brainstem isn't really so much about conscious detection of sweet and sour, salty, bitter tastes, but as it turns out, when the body craves certain things like maybe we're craving something that's high in fat, that is fat, of course, is calorically dense, and that's a specific kind of signal that can activate the brainstem to suggest that you've just consumed some things that are very calorically dense, so maybe that's going to make you feel full a little bit faster, because the digestive system can actually taste. This is a weird one, right? There are chemical receptors like the taste buds all throughout the digestive system, and they can then respond to the chemical contents of the food. And so it's a form of taste, but it's not taste that we're consciously aware of, but it's certainly a form of chemosensation that plays a role in signaling to the brainstem the nature of the foods that are present in the stomach and the intestines and their caloric density, right? At a higher brain level, the hypothalamus, we keep talking about that, right? So we know that the hypothalamus is connected to the pituitary gland. The pituitary gland is the master gland of the endocrine system. And so here we see the posterior pituitary in particular is where we're getting input from the hypothalamus, whereas the anterior pituitary is where a lot of hormones are manufactured. And then, of course, through that, the pituitary releases these hormones, which then travel to these peripheral organs of the body, either influencing our organs or influencing other glands of the endocrine system, whether it's the sex glands or the adrenal glands or anything like that to produce different hormones. So we're going to subdivide the hypothalamus a little bit here. The hypothalamus is really a bundle of different nuclei. And we've talked previously about things like the suprachiasmatic nucleus that regulates our circadian rhythms. Here, other rhythmic behaviors. Eating is, in fact, a rhythmic behavior. It's just not a circadian rhythm. It doesn't occur on a day-night cycle because it occurs several times throughout the course of a single day. So it's more of an hourly type kind of rhythm, but it is a rhythmic behavior. So we see this as a common theme of these kind of motivated hypothalamic-driven behaviors is that they're all in various ways rhythmic, right? In this case, the lateral hypothalamus is a nucleus that sits more towards the sides of the hypothalamus, clearly, that activates as a hunger-on signal. What we know is that if we directly stimulate this artificially with electrodes, it increases, say, rats to eat more, right? Makes them feel hungry, apparently, and therefore they eat more. If we lesion it so that it is no longer possible to stimulate this part of the rat's hypothalamus, they simply just stop eating, right? And something else that we see is that the LH also sends signals to the VMH. That stands for the ventromedial hypothalamus. So now this is more towards to the belly middle side of the hypothalamus. This is actually a separate thing that's called the hunger off part of the brain. So if we activate the ventromedial hypothalamus, that also causes the stopping of eating behavior, cessation of eating. There's clearly interactions between the LH and the VMH. So if I say that eating is a form of regulatory behavior, what are we trying to regulate? Well, as it turns out, what we're trying to regulate is we're trying to regulate energy reserves in the body. And we need different kinds of energy reserves available over different time span. So I have listed here immediate, short-term, and long-term. Immediate is what we need right now. Right? So in the moment right now where I'm moving, I'm waving my hands around, I'm talking, that requires muscle contraction. The muscles contracting need energy. That comes from glucose and oxygen, right? So if you remember some basic biology, glucose and oxygen combine through a process known as phosphorylation to create ATP, and that's what keeps your cells alive. For the muscles, it may work a little differently. There's also anaerobic metabolism that can also feed the muscles as well. But in general, though, you're going to need some glucose for that. Right, so that means we have to have some glucose freely floating in the bloodstream and available for those immediate metabolic needs. We don't want that to be too high or too low, right? Just like with blood pressure, we don't want it to be too high or too low. We don't want hyperglycemia. We don't want hypoglycemia. We want it somewhere in the middle, right? Some predetermined set point that is in what we call a medically defined healthy range, right? But we don't want to just have that, because what happens if we use it up really quickly? I might take off running down the street, and then I use up all of my glucose in my bloodstream. How do I replenish it? Well, I could eat, of course, but that takes sometimes minutes to hours to restore blood glucose levels. And that can lead to an extended period of hypoglycemia if I have to wait for eating to occur and then digestion to occur to get everything back to the normal state. So what I have to do here is I have to have a short-term storage so that I can replenish the blood glucose as needed. And this is where the liver is involved. The liver stores energy, at least one of its many jobs. And so what happens here is that some of the excess blood sugar in our bloodstream is converted into glycogen, and the hormone insulin plays a role here, produced by the pancreas. And so now the glycogen is stored in the liver, and when blood sugar levels begin to drop as more and more time goes by after I have eaten, and gradually blood sugar levels begin to decline, or if I increase my physical activity to some extent, and that even more rapidly begins to deplete the immediate blood glucose, then the liver just replenishes that, right? And just gradually is releasing that glycogen back into the bloodstream to be converted back into glucose. Or if in the case where, like I said, if I just pick up and I start running down the street, that's going to quickly use up my glucose. But again, the liver can just spike the bloodstream with a very rapid release of glycogen. So this can happen fairly quickly. It's kept and held in the liver as a storage mechanism, but it can be fairly easily and quickly converted back into glucose when that is needed, right? But there's a separate long-term storage. The idea here is that evolution has equipped humans with the ability to store energy as body fat, right? Because of course there might be times where, like I said, once our blood sugar levels drop, and then maybe even the glycogen storage in the liver drops, we're going to need to replenish that energy pretty soon, which means we need to eat. But not only does that take time, there could be periods where there is not a good, consistent supply of food available to us. And if we go too long without eating, of course, we're going to possibly die. So that means that we need to have some way of storing for those lean periods, right? Storing for the winter, perhaps, when there's less food. So in that case, body fat. And so now, like there is with an ideal sort of sense of blood sugar levels, there is an ideal body fat percentage, right? You don't want too low, you don't want too high. There are, again, health consequences of both. So there is some sort of predetermined set point for where the body fat storage ought to be. So in order to create body fat, now the excess glucose is converted into another chemical, glycerol, that combines with fatty acids to make triglycerides, and then the triglycerides are used to form adipose tissue, which is body fat, right? Now, body fat can also be broken back down to blood sugar through a long convoluted process. Some of it can also be directly metabolized by cells of the body as well. But the problem, though, again, is that this is a fairly slow process, and the breakdown of body fat back into metabolizable constituents takes hours and tends to only happen when we're sleeping, really, for the most part. So that means that body fat is not really used to replenish blood glucose in the short term, right? Like if I need energy right now to run down the street, the breakdown of body fat is not going to help me out very much for that, right? Body fat is really just slowly breaking down to compensate for someone who is over some long period of time eating less than their body needs over some period of time. And that, of course, typically happens in times of a food shortage. I have a little small note here, for example, that glucose is pretty important for the function of the brain. The neurons really rely on glucose for their metabolic function, right? Less than oxygen, really, it's just glucose. So that means that in addition to supporting muscle contraction, having an appropriate level of blood sugar is also important to support brain function. And when our blood sugar drops, our cognitive function declines pretty dramatically. So that's another important thing to think about in the context of all of this. So what are the actual mechanisms here? Well, when our blood sugar, as I said, slowly drops, the longer it's been since we have eaten, the declining blood sugar is detected, and this results in the parasympathetic nervous system in the gut producing a hormone called ghrelin. Ghrelin is also even triggered by an empty stomach, right? But it's also more about that low blood sugar. And that's going to then activate the brainstem and the hypothalamus. We're going to activate that lateral hypothalamus and get the hunger on response. So we're going to start eating, right? Then the next question is, when do we feel full or satiated, right? How do we trigger a hunger off signal? So there's a variety of hormones that are listed here as shown, but there's also, as I had said, a lot of cognitive factors here. Sometimes people override their hormone signals and overeat, perhaps on purpose, because sometimes, we have cultural practices like eating 3 square meals a day, breakfast, lunch, and dinner. So that may mean that we will eat those 3 meals even when we don't feel hungry, because it's just been a culturally reinforced practice that you're supposed to. Or you might have been trained to eat everything that you were served, to clean your plate. So again, these kinds of acquired behaviors sometimes will override these hormonal signals. And then there are the sensory things. So again, with gustation, we might be driven to eat certain things, but also sometimes we overeat out of pleasure because, you might be full after dinner, but because there's a dessert that you really want to eat because you just really like it, you may eat it anyway because you want to, right? So because that's, again, a sensory driven, pleasurable process. So the hormones here, though, first one up is cholecystokinin, CCK for short, because I don't think we want to say cholecystokinin over and over again, right? So, but this is when I said the small intestine begins to fill up, right? So there's stretch receptors, this is part of interoception, right, in the small intestine. And as it begins to fill with food, slowly, of course, right? Because, you eat, food goes into your stomach, slowly the stomach empties into the small intestine. And as that begins to fill up, then CCK levels begin to rise in proportion to the volume of food in the small intestine. This is going to deactivate the hunger on part of the hypothalamus, right? So it just deactivates the lateral hypothalamus. PYY does very much the same thing, except the strength of the PYY signal is a little different than CCK. CCK, as I said, the strength is in proportion to the volume of the food. PYY is in proportion to the caloric density of the food. So when you have a high calorie meal, now we're going to get more PYY. It could be low food volume, right? So a spoonful of sugar or a spoonful of oil or fat is very small in volumetric terms, but it is very calorically dense. So that causes an increase in PYY, even though the CCK signal might be small, right? Conversely, of course, if you have a high volume of low caloric food, like a bunch of lettuce, then the CCK signal might be stronger, but the PYY signal is weak. So some combination of high volume, high calorie food gets you a stronger effect here to increase satiety from both of these hormones together. Now, this takes a little bit of time because, of course, it takes a little bit of time for the stomach to empty its contents into the small intestine. And what takes an even slightly longer period of time is the role of insulin in this, right? So separate from insulin helping play a role in storing the excess glucose into the liver and body fat, it also influences the brain and acts as a hunger off signal as well. So what's happening here, though, is as the blood sugar level rises, and that's a very gradual thing over the course of after we're eating and slowly digesting the meal, then gradually insulin rises, and over the course of several minutes to even 30 minutes or more, insulin levels continue to rise, and that then eventually deactivates that hunger signal, and we get that hunger off signal from, in particular, the VMH in this case, right? This is directly affecting the VMH to stimulate that hunger off part of the brain. And then even beyond that, now another hormone produced by our body fat, leptin, has this effect. And what it does is it actually interacts with CCK, and it makes the CCK signal to the brain stronger. So that means that because CCK is already influenced by the volume of the food you eat, you can eat a small volume of food, but if you have high leptin levels, leptin will help boost that CCK signal that makes you feel full even faster than you otherwise would. This provides evidence now of this fat storage regulatory mechanism, because why would we be motivated to eat less if we already have a lot of body fat? Because we don't want to have too much body fat. And so the role of leptin is to help regulate eating behaviors, and in general, it reduces hunger as our body fat levels go up. But lower levels of leptin, if our body fat levels go down and our leptin levels are down, then of course that would tend to actually increase hunger, because then the CCK signal is no longer being boosted by the leptin. The exact role of leptin in regulating body fat has been explored with this mouse research. You can kind of see in my background here, I've got the pictures of the mice here. So what we can do is we can identify a gene called the OB gene, and this gene regulates leptin production. And we can use a knockout procedure to disable that gene in mice. And so now we'll call this the OB mouse, because of the OB gene has been knocked out, and this mouse no longer produces any leptin. And what we find, of course, is that because it doesn't, no matter how much weight it gains, there's no leptin, this mouse will just overeat, because that CCK signal is not really being aided in any way. And so you see the mouse in the background on the left there, it's just kind of blowing up and getting really big. Now on the right-hand side, this is actually the same mouse at a different time, where after it begins to receive leptin injections, it starts to lose weight, right? Because now, of course, these injections are making up for the genetically missing leptin, and now it starts to better regulate its eating behavior, and it begins to lose weight. So now we see later the mouse has dropped some ounces there. Now you might think, oh, does this mean that we can use leptin injections to treat obesity in humans? And as it turns out, the answer is no, because humans with obesity issues are not experiencing that because of some genetic mechanism that causes them to insufficiently produce leptin. Instead, it seems to be much more of a resistance level, that the hypothalamus in the brainstem, where we find those receptors on their dendrites. So again, dendrites have these receptors that are unlocked by neurotransmitters and or hormones, which are the same thing. But neurons, we know metabolically, can either increase or decrease the receptors on the surfaces of their dendrites. And we learned that when we talked about neuroplasticity and Hebb's law and stuff like that. And so what we're seeing here is that chronically elevated leptin levels, as well as chronically elevated insulin levels that come from high blood sugar and high body fat, eventually result in a loss of receptors on the dendrites of all of the relevant neurons in the brainstem and hypothalamus. That makes those neurons now less sensitive to or resistant to the signal of these hormones. And then, of course, the result is that these hormones no longer are effective. So injecting those hormones to create more of them isn't really going to change the fact that the brain stops responding to them. We'd have to find a way to maybe get those neurons to grow more receptors for that, but that's not easily remedied, right? Okay, that's where we're going to call it for this particular half of the chapter, and we will then create another video for the emotion half.